Hello, this is Mrs. Shostak. I just finished reviewing the Module 3 quiz and going through the item analysis. And I noticed that there were 3 questions in particular that the group seemed to have trouble with. I would like to give you an opportunity to earn some additional credit by showing mastery of these concepts. So I'm going to post them on a discussion board. It's going to be titled Mastery Credit Module 3. And if you review these slides and then post a brief one-line describing the correct answer to the questions you missed, I'll give you partial credit for those questions. You can also reach out to me and ask me for further clarification. So I'm going to go through these really quickly. Let's see. So again, you're going to go to the discussion board and you're going to put a brief description of what the correct answer is for the questions that you missed. I'll review those and provide you with partial credit. So the one that people really seem to have trouble with is guidance versus regulation. And a lot of folks said that guidance documents are legally binding and require compliance. But the reality is the regulation is legally binding. The guidance document is a recommendation by the regulatory agency. You are not legally required to comply with guidances, but you are legally required to comply with regulations. And it's important that you know the difference between the two. So guidance documents are recommendations, not legally binding, and regulations are legally binding. So I'm going to play this again for you.
So what is clinical data management? Our textbook states that clinical data management is the work performed on data from a clinical trial, from the preparation to collect that data through the time it's extracted for final analysis. Clinical data encompasses all the data generated by and related to the conduct of a clinical trial. Examples of clinical data include medical history, vital signs, physical exam, laboratory data, and adverse event data. The goal of clinical data management is to produce complete, reliable data that can be used to answer the question posed in the protocol. The clinical data management team is responsible for ensuring that clinical trial data are integrated into a format that can support the analysis. Regulations govern what we do. All data management activities have to comply with the general standards of good clinical practice. But in addition, there are a few regulations and guidance documents that specifically address clinical data management activities. One that you're going to hear very frequently is 21 CFR Part 11, and CFR stands for the Code of Federal Regulations. This documents FDA regulations governing electronic data records and electronic signatures. 21 Part 11 tells us that we have to validate databases to ensure they're accurate and reliable, that we have to control access to them, and that we have to have a computer-generated audit trail that documents the creation, modification, or deletion of electronic records. Now remember, 21 Part 11 is a regulation. This means compliance with it is required, not just suggested. There are also several guidance documents that we're going to have you read over. One is the Guidance for Industry in relation to electronic source data and clinical investigations. Another is the Guidance for Industry Computerized Systems used in clinical trials. And then we also have the ICH GCP Guidance E6. Section 5.51 through 5.512 relates directly to clinical data management activities. There are some other places where we look for best practice. One of these is the Society for Clinical Data Management, or SCDM.
So another question that a lot of people missed was error rates. The question I had up is, do the FDA and ICH regs dictate the acceptable error rate? And the answer is no. There are no regulations that say what an acceptable error rate is. However, there are some commonly accepted industry standards. So again, this is the difference between what is required per regulation and what is industry practice or best practice. And it's important to know the difference between a regulation that's a requirement and a best practice that's just the guidance of the industry.
So how do we know what quality data is? There's no regulatory definition of quality data. In general, the drug development industry expects that quality data is going to be fit for purpose, it's going to be complete, it's going to be valid, and it will have an acceptable error rate. Fit for purpose means the data can be used in the analysis and decision-making process. You have the data you need to make the decision. In other words, that data is going to allow you to answer the questions outlined in the protocol. Data is considered complete if all the expected attributes of the data are present. For example, are all start and stop dates for an adverse event present in the database? Valid data is data that is correct and truly represents the values they're intended to measure or reflect. There's no regulatory definition for an acceptable error rate, but in general, the drug development industry considers an acceptable error rate to be between 0.1% and 0.5%, and that roughly means that you are finding 10 to 50 errors per 10,000 fields.
Another question that people missed a lot was key variables. So key variables are essentially the
Primary data points that you're going to use for analysis. And you can find those in the protocol and in the analysis plan, but also in the protocol synopsis. And this is actually part of what we went through when we were reviewing the protocol synopsis for our little mock trial. So the correct answer would be all three of these items.
Let's talk a little further about the concept of key variables. Key variables are essentially the variables or data that you are going to be using to prove or evaluate your product's safety and efficacy. They have to be present and they have to be correct in order for you to get a valid analysis of what you are claiming this drug can do and what it actually does. Where do you find the key variables? Well, the protocol and the analysis plan will define the primary and secondary efficacy outcome measures. You can see I have a little example up here that you can take a look at, and we'll talk about this further on in the course when we are developing our own project-specific plans. The safety variables